Hi, my name is Ellen, and the title of today's paper analysis is Extensive Age-Dependent Loss of Antibody Diversity in Naturally Short-Lived Turquoise Killifish. So what this paper essentially talks about is the killifish, which is the model of aging, vertebrate aging to be specific, rapidly loses its antibody diversity as it ages. This study was carried out by the famous killifish researcher Dario and was carried out in Max Planck Institute for Biology of Aging and was published in eLife in 2022. Before starting of today's paper analysis, I want to give you a brief background about aging immune system. Our immune system works less efficiently as we age, and we all know this. As you can see in the figure on the right, the aging immune system, or immunosenescence, is caused by various problems of aging. One example is thymic involution, or the gradual loss of the organ called thymus, which is really important for T cell production. And T cells are important for protection against infection as well as cancer. Another example is altered gut microbiota. In fact, our 70% of immune system resides in the gut, forming an intimate connection with these bacteria. And when you have less diverse presence of bacteria or negatively affecting bacteria in your system, you're likely to be getting a high infection risk and poor vaccination responses. We could empirically see this in the COVID-19 pandemic. So this is the figure that I borrowed from one of the statistics that derived from COVID-19 pandemic. And as you can see, for the aged population, there are not much people with the COVID-19 cases. However, for the death, the population of the aged individual is pronounced. So old individuals are more susceptible to infection and less responsive to a vaccine, as evidenced by COVID-19 statistics. However, an in-depth time-lapse study of immune aging in humans is infeasible. So a model organism is wanted. And this is where the killifish comes in. The killifish is a short-lived organism gaining its attention as the model of vertebrate aging. A specific strain called GRZ strain, for example, has a median lifespan of only 4 months or around 120 days, proving its efficiency as a model of aging. Killifish can generate humoral immunity, producing antibodies, and killifish enables the study of other bodily tissues exclusive of blood, like humans. As for the key points of this paper, first, the study is the first to perform immune repertoire aging in turquoise killifish. Second, older killifish have reduced diversity of antibody repertoire. Third, blood and gut immune repertoire have disproportionate patterns of loss by age. As for the methods, they did targeted RNA sequencing of immunoglobulin or antibody heavy chain. They did 2 separate experiments. First is the RNA sequence from whole body, where they divided the groups into 4: juvenile, young adult, adult, and elderly. And they did a separate experiment for the gut, where they divided the groups into 2: young adults and elderly. This is the results of the study. The first experiment was done on the whole body or blood. Figure B is showing you the differences in alpha diversity between age groups. And what alpha diversity tells you is within individual variability of antibody repertoire. The x-axis is the diversity order, or the Q value of any particular B cell clone. The low Q value B cell clones are the ones that are called small B cell clones, only composed of naive B cell clones, naive B cells that are not much proliferative and are not activated, as opposed to the small ones. The ones with larger Q value are called the large B cell clones, which are more activated and more proliferative. And these are the ones that are important for the recurrent infection or cancer protection. And the y-axis is showing you the actual alpha diversity values. And in the figure you can see that the smaller B cell clones are not much affected by the age, whereas the larger B cell clones, important for the protection against infection and cancer, are much affected. As for the figure H, this is telling you the beta diversity, or the between individual variability in alpha diversity. In repertoire diversity. So, and the beta diversity was measured by the pairwise RDI distance, which stands for the repertoire diversity index. This is essentially an average root mean squared value. So as the killifish age, the between individual diversity increases. And the similar experiment was done on the gut samples, and you can see that the effect is more pronounced in terms of alpha diversity, and quite similar, or you could say a bit more pronounced for beta diversity. So taken together, aged fishes have a reduced diversity or alpha diversity of antibodies within themselves. This is presumably due to loss of healthy immune function by aging, and rapid loss of only large activated clones and minor loss in overall composition was shown for blood, but not for the gut. Aged fishes have a greater variety of antibodies between themselves, or beta diversity, than younger ones. And this is presumably due to the individual difference in history of life, for example, diet and infection. And this is quite suspicious since the fishes, I presume, are grown in the same vessel or same kind of small space, and I don't really think there would be a difference in diet and infection between these samples or individuals or killifishes. Anyways, the last point is that the effects of repertoire aging is more pronounced at gut, as I told you, a mucosal surface. And this makes sense because the gut is more intimate with the external surface, with the gut microbiota, as I told you in the background. And blood is kind of indirect results of this. And that was it for the paper. So the take-home messages for this paper are as follows. The killifish is a powerful model for studying immunosenescence longitudinally. And this study was the first to prove this. And of course, the mucosal immunity was more severely affected by age than peripheral immunity, presumably due to the altered gut microbiota. Lastly, age-related alterations in adaptive immune response may be responsible for severity to infection, for example COVID-19, and future studies are warranted. Thank you.